Center for the Ethiopian Educational Information and Communication Technology presents Educational Satellite Television Programs.
Hello students, this is Asrat. Last week we dealt with cellular respiration. In our lesson we tried to see the four major stages of cellular respiration. I hope you all remember all those four stages. The first one was about glycolysis, and the next one is about link reaction, and the third stage of cellular respiration is said to be Krebs cycle, or we call it citric acid cycle, and the fourth stage of cellular respiration is going to be electron transport chain. We also call it, in addition, chemiosmosis. So cellular respiration takes place where, as you know, it is a major site for cellular respiration is a mitochondria, and this mitochondria is said to be the powerhouse of the cell, as it produces the energy currency of the cell that is delivered throughout the cell for every activity that requires energy. Let's go to the lesson that we are going to have today. So our third stage of cellular respiration is about Krebs cycle. It is named after the scientist Krebs. Okay, Christian Hans Krebs, and we also see electron transport chain following that. So, in our previous video, you have studied about glycolysis, the first stage. You all remember that glycolysis takes place in the cytoplasm. It is an aerobic stage of cellular respiration. Do you remember that? And it is also a process that breaks down glucose into two smaller molecules that can easily pass through the mitochondrial membrane that are called pyruvate. They are 3 carbon molecules. In the process, there will be 2 net ATP produced out of a total 4, because 2 of the ATP molecules already consumed in the starting process of cellular respiration. And there will be also reduction of NAD, so there are 2 NADs as well reduced in the process. This pyruvate now can pass through the mitochondrial membrane because it can easily pass. It is smaller, and it is also able to pass through the channel proteins. But glucose can't, because it is bigger and there is no channel protein to allow it to pass through the mitochondrial membrane. This pyruvate then passes to the mitochondria, as I mentioned, and the next stage takes place called link reaction. In the link reaction, these 2 pyruvate molecules produced here will combine with, as you can see, CoA. We call it coenzyme A. It's a bigger molecule that carries the pyruvate to the next level, and carboxylase, acetyl coenzyme is produced, In the process, NAD is reduced, and there is also carboxylation. We call it decarboxylation. Carbon dioxide is released. Don't forget, 1 carbon dioxide is now removed from a glucose molecule. So, now we go to our lesson called Krebs cycle. We also called it citric acid cycle. In this process, there are very important things happening. One of the events that happen is the acetyl coenzyme molecule, which is actually produced from the link reaction, is now passed to this stage and combines with a 4-carbon molecule already waiting inside the mitochondrial matrix. You should know Krebs cycle and link reaction take place in the matrix of the mitochondria. This molecule called oxaloacetate is waiting for it. Then acetyl coenzyme A combines with oxaloacetate, and this coenzyme A returns back to bring another pyruvate, which is left already, because you remember one pyruvate still is there, and citrate is formed. That's why we call it a citric acid cycle. And this citrate undergoes a cycle of decarboxylation, changing from 6-carbon molecule to 5-carbon molecule, from 5-carbon molecule to 4-carbon molecule, until it regenerates the original molecule called oxaloacetate. Let's see the cyclic process here. Here is the very important cyclic process of citric acid cycle or Krebs cycle. You can see that carbon dioxide, coenzyme A and acetyl molecule are coming into this stage. You can see that oxaloacetate is waiting for them, a 4-carbon molecule, combines with oxaloacetate, and this coenzyme returns back to bring another coenzyme, while this oxaloacetate forms a 6-carbon molecule, and there is decarboxylation of that citrate. 1 carbon is added now. How many carbon do you remember in pyruvate molecule? There were 3. 1 carbon dioxide is removed during link reaction. Here is another one, 2nd carbon. And again, this 5-carbon molecule, we call it succinate. And this succinate now removes 1 carbon dioxide. In the process, NAD is reduced. ATP is also formed. Consider how many NAD is reduced, how many ATP is formed in the process, and how many carbon dioxide is released. Then the 4-carbon molecule, we call it alpha-ketoglutarate, and it goes to fumarate, malate, then oxaloacetate molecule. You see, oxaloacetate is regenerated. All the carbon dioxide that is carried by pyruvate or acetyl molecule is released. 2 carbon, 1 carbon in the link reaction. Now 3 carbon dioxide molecules are released. So this is the stage of Krebs cycle, where most of the hydrogen molecule is carried by NAD, FAD, and another NAD molecule. And there is also another NAD here. How many NAD molecules do you see here? 1, 2, 3 NAD molecules are reduced in one Krebs cycle. Then there is also another Krebs cycle for a single glucose molecule, which again produces another 3 NAD and 1 FAD, generally making 6 NAD molecules to be reduced, 2 FAD molecules to be reduced, and go to the next stage of cellular respiration, which is the most important stage of cellular respiration, to produce 32 ATP molecules. You can see that so far we produced very less amount of ATP molecule. In Krebs cycle, there are only 1 ATP. In the next Krebs cycle, from 1 glucose molecule, totally 2 ATPs are made. And this is what we call substrate level phosphorylation. So let's pass to the next stage of cellular respiration. Before that, but I need you to remember very important points in Krebs cycle. 1, energy enters in the cycle in the form of 2 carbon molecule called acetyl-CoA. This CoA, as you know, is going to return back to bring another acetyl molecule. It is recycled in the process. Then oxalic acid or oxaloacetate combines with acetyl, forming citric acid. In the cycle, 1 ATP, 3 NAD, and 1 FAD molecules are produced. 2 carbon dioxide molecules are released. And, of course, Krebs cycle link reaction are happening in the matrix of the mitochondria in the presence of oxygen. So let's go to the next stage of cellular respiration, called electron transport chain. We also add chemiosmosis in the process, because it is the process that releases the most of the ATP released from glucose. In the electron transport chain, you might just get confused about it. It contains, or it happens in the cristae of mitochondria. Cristae means the inner folded membranes of mitochondria. And the mitochondrial layer inside is very highly folded in order to increase the surface area for this process. So we are going to see a very slight portion of that cristae and how this electron transport chain produces the energy. And it contains these complex proteins that are used to transfer electrons from one electron acceptor to donor to another electron acceptor through a redox reaction. In the process, there will be some molecules that allow the proton to be pumped across the cristae or across the mitochondrial membrane so that it releases most of the energy it requires. For example, this is just a picture of the electron transport chain happening in the mitochondrial membrane. As you can see, this is the inner membrane space. This is the outer membrane space of the mitochondria. And you can see this membrane made of just like phospholipids. And this membrane has some very important proton pumps in it. These proton pumps in it will help to pump the hydrogen ion out in the process, and then the electrons jump from one electron molecule, transferring molecule to the next, and then the next, and the next, until that electron combines with hydrogen ion to form the end product called water in the cellular respiration. So let's see the stages in detail. What happens first? Okay? The hydrogen atom carried by reduced NAD molecules. How many are they from a single glucose? Students? Okay, let me tell you. In glycolysis, 2 NAD. In link reaction, again, 2 NAD. How many? 4. And in the Krebs cycle, 2 Krebs cycle happens in the process, 6 NAD. Totally 10 NAD molecules are reduced in one single cellular respiration process using one glucose molecule. These reduced NAD molecules, of course, there is also fat, don't forget it in Krebs cycle, 2 fats, they actually are released and split into protons. This hydrogen comes in the process as I have shown you in the picture. That actually comes here. Look, these reduced NAD molecules come here, and this hydrogen brought splits into electron and proton. As this electron is taken to the other electron acceptor, ubiquinone, okay? Then to cytochrome complexes, then to the other electron acceptors. As this electron jumps from one electron acceptor to the other, the energy released here will be able to help pump this hydrogen out of the outer mitochondrial membrane to the inner mitochondrial membrane. In this process, look what happens is 3 hydrogen pumps. The reduced NAD dehydrogenase complex, the ubiquinone, and the cytochrome complexes, primarily containing iron, all of them help in pumping hydrogen into the inner space, making the hydrogen ion to concentrate on this part of the mitochondria, eventually leading that hydrogen to be pumped back through ATP synthase. This is an enzyme that helps us in the synthesis of ATP through chemiosmosis. Chemiosmosis. You remember osmosis and chemi, right? Chemical osmosis. The hydrogen ion accumulated in the inner membrane, brought by reduced NAD and fat molecules, will be pumped into the inner membrane. Then they are finally accumulated here, and a concentration gradient is formed. That concentration gradient will be able to pump it back. As it pumps, it spins the rotor that helps to make ATP molecules. And from one hydrogen pump, 3 ATP from NAD, and there are 10 from NAD, around 30 ATPs are made. And fat molecule, 2 fat molecules, when they are actually transferred, 2 ATP from each fat will finally produce 4 ATPs, making the total ATP made through the chemiosmosis 34 ATPs. I would like to summarize this with a very short sentence. In electron transport chain, very complex events happen. Reduced NAD and FAD molecules are brought to the electron transport chain. And this complex of molecules that are found in the membranes of the mitochondria will receive the hydrogen atom. This hydrogen atom splits into electron and proton. That proton is pumped by these three important proton pumps: reduced NAD dehydrogenase, ubiquinone, cytochrome complexes. These three pumps will help to pump the hydrogen as it comes from NAD and FAD, and helping it to be passed to the intermembrane of mitochondria, finally pumped back to the outer membrane. What happens when it comes down there? Let me show you the picture again. Here what happens the event. Look the event. The proton, 6 protons coming from one pyruvate are pumped down here. And as this hydrogen pumps, the electron is coming through the cytochromes, will be combined with the final electron acceptor of cellular respiration, oxygen, is waiting there. And this oxygen combines with the hydrogen sent from the ATP synthase, forming water as the end product of cellular respiration. At the same time, what happens is this oxygen will be used to form water as the end product of cellular respiration. So I need you to remember those things. Electron transport chain is said to be oxidative phosphorylation, as it uses oxygen to finally receive the electron and the proton forming water. And it is this stage that makes the highest amount of ATP molecules in cellular respiration. Let's go to the next. Okay? This is the electron transport chain. We have already seen this part, ATP synthase, and how the protons are pumped in the previous diagram. Generally, this happens. In a nutshell, you can see the whole stage of cellular respiration. Glycolysis, glucose, 2 ATP molecules are made as glucose breaks down to pyruvate. This pyruvate now can pass to mitochondria, forming acetyl coenzyme A. You know that coenzyme A is already there, combines with acetyl as decarboxylation took place. This acetyl passes to Krebs cycle, producing 2 ATP in general from 2 Krebs cycles, and as well, many decarboxylation took place and reduction of NAD. Electron transport chain produces 32, sometimes 34. Totally, it becomes 36 ATP molecules are made from aerobic respiration, which happens in our cells. But what if sometimes the level of oxygen is very low in our body, or sometimes there are some living organisms that cannot use oxygen fundamentally, so they use other ways of producing energy. That kind of cellular respiration, we call it anaerobic respiration, or we call it anaerobic pathway of cellular respiration. What happens during this stage is there is no oxygen to use. Most importantly, if you put yeast in alcoholic industries, if you see yeast in alcoholic industries, it is limited from getting oxygen. So it uses a different way of producing energy for survival. Okay? So if there is no oxygen present, the final reaction of oxidative phosphorylation will not be there, which is electron transport chain. If oxidative phosphorylation is not there, there is no need of oxygen in the process. So what happens as a result, this comes to halt, ATP synthase will not produce energy or will not be undergoing chemiosmosis. There is no reduction of NAD or FAD in the process. finally what happens is Krebs cycle starts to stop. And the link reaction also will not be able to take place. Why? Because link reaction, Krebs cycle, and electron transport chain, they all take place in the mitochondria, and they all take place in the presence of oxygen. So all these stages, step by step, from electron transport chain to Krebs cycle to link reaction, they will be halted. And there is no reduction of NAD and fat in the process. Only glycolysis can happen if the conditions are anaerobic. Let's see how that happens. In this process, you can see that glucose is changed to pyruvate. This is simple because it doesn't take place in the mitochondria and it doesn't need oxygen. In the process, you can see that totally 4 ATPs are made and a net of 2 ATP molecules are produced, because you know that 2 ATP molecules are already consumed in the process. And again, here you can see that NAD is reduced. But how can we return or oxidize this NAD back again? For that, cells usually use the other different mechanism. In order to recover NAD back to its oxidative stage or oxidized stage, it will be changed to 2 different kind of molecules so that this NAD, which is reduced, returns back to its normal state. For example, in the anaerobic pathway, glucose changes to 2 pyruvic acids and 2 lactate molecules are produced in our muscles, if you remember when you are doing vigorous exercise. And the other one is when 2 pyruvate molecules are changed to alcohol in case of yeast, in which case carbon dioxide is made. At the same time, this NAD, which was reduced, recovers back to its original state, means becomes oxidized. There is electron accepted in the process, so that in this way, cells undergo anaerobic pathways. Most importantly, yeast makes alcohol through this pathway. You know this since you have studied that in grade 9. And the other one is lactate, is produced during vigorous exercise of human and other animals. And this anaerobic pathway is used for many economic activities in manufacturing of different kind of chemicals. Sometimes it is beneficial, though it produces very less amount of ATP. You know that aerobic pathway produces 36 ATP, but this one totally produces only 2 net ATP, and it leaves the body with lots of residual molecules, which might be wastes. Lactate, for example, is a waste or residual. Sometimes our body recovers it, sometimes removes it as a waste through our urine. Glucose, after it changed to pyruvic acid, there are so many microbes that can use this pyruvic acid to change it into our useful products. If we put it in industrial application and transform these bacteria into a producer, we can produce several kind of products that can be useful to our body, to our diet, or to our health. Cheese, for example, is made by propionic bacteria. This propionic bacteria releases carbon dioxide from pyruvate and makes Swiss cheese. The other one, Bacillus, Lactobacillus, and the Streptococcus bacteria are able to make lactic acid that are used in many different kind of products we use in our life. Yeast, as you know, makes alcoholic drinks. Clostridium is used to make acetone and isopropanol products that are used in nail polishing, rubbing alcoholic brushes. Escherichia coli and Acetobacter help us to produce acetic acid. We use these chemicals for many industrial activities. So anaerobic pathway is useful in these ways. So I need you to differentiate the two. Anaerobic pathway will help to produce products, while aerobic pathway produces large amount of ATP molecules. I want you to compare 2 very important runners. Usain Bolt, the fastest runner of the world, and Haile Gebrselassie, our legend runner How do you think they too use while running in the race? Do you think they use aerobic or anaerobic pathway? I want you to answer those questions by yourself. Who is the fastest and who needs oxygen so urgently? And if oxygen is not there, how do you think Usain Bolt would produce energy within that 9 seconds? Why Haile Gebrselassie runs marathon for 2 hours? Who do you think breathes aerobically and produces energy aerobically? And who do you think produces energy anaerobically? That is the answer. You'll tell me later. Good day. Thank you.
How do you turn that bite of food into a chemical that a cell can recognize and use as energy? The first step is altering the food into its component chemical compounds and then getting those molecules into your cells. That process is called digestion. Once inside your cells, the process of turning that bite of food into useful energy by cellular respiration begins. The process of digestion results with carbohydrates and other molecules being removed from the consumed food and transported into the bloodstream. From there, nutrients, like the carbohydrate glucose, will leave the bloodstream through a capillary wall and enter a tissue cell. Once inside the cell, cellular respiration will completely oxidize the glucose molecule, releasing high-energy electrons. The overall goal is to make ATP, a storage form of energy for most cells. Cellular respiration is a 4-stage process that begins with glycolysis. Glycolysis literally means splitting sugars, and it is the first step of cellular respiration, occurring in the cytoplasm of the cell. Glycolysis consists of 2 distinct phases, an energy investment phase and an energy harvesting phase. In the energy investment phase, 2 ATP molecules transfer energy to the glucose molecule, forming a 6-carbon sugar diphosphate molecule. This molecule splits, and the energy harvesting phase begins. During this phase, the 2 3-carbon molecules are converted to pyruvate, and ATP is formed. Glycolysis is a 10-step reaction that involves the activity of multiple enzymes and enzyme assistance. In the process, a net of 2 molecules of ATP, 2 molecules of pyruvate, and 2 high-energy electron-carrying molecules of NADH are produced. When oxygen is present, the pyruvate molecules and NADH enter the mitochondria, and the next stage of cellular respiration begins. The next stage of cellular respiration involves the movement of pyruvate into the mitochondria, where it undergoes oxidation. Each pyruvate molecule is converted into a compound called acetyl-CoA. In the process of pyruvate oxidation, electrons are transferred to NAD, producing NADH, and a carbon is lost, forming carbon dioxide. The next stage is the citric acid cycle, also called the Krebs cycle. Here, acetyl-CoA will bind with a starting compound called oxaloacetate, and through a series of enzymatic redox reactions, all carbons, hydrogens, and oxygens in pyruvate ultimately end up as carbon dioxide and water. The pathway is called a cycle because oxaloacetate is the starting and ending compound of the pathway. For every glucose that enters glycolysis, the cycle completes twice, once for each molecule of pyruvate that entered the mitochondria. During pyruvate oxidation and the citric acid cycle, a net of 8 NADH, 2 FADH2, 2 ATP, and 6 CO2 are produced for each glucose molecule. In order to understand how the majority of the energy is produced by aerobic respiration, we need to follow the NADH and FADH2 molecules to the next stage, the electron transport chain. The electron transport chain is a series of membrane-bound carriers in the mitochondria that pass electrons from one to another. As the electrons are transferred between the membrane proteins, the cell is able to capture energy and use it to produce ATP molecules. Proteins in the chain pump hydrogen ions across a membrane. When the hydrogen ions flow back across the membrane through an ATP synthase complex, ATP is synthesized by the enzyme ATP synthase. Oxygen acts as the terminal electron acceptor. By accepting electrons, oxygen is reduced to form water, a byproduct of the electron transport chain. All the high-energy electron carriers from the previous stages of cellular respiration bring their electrons into the chain. From this, the bulk of ATP from the entirety of cellular respiration is produced, a net of 32 to 36 ATP. In summary, we have seen how the 4 stages of cellular respiration are responsible for converting the energy found in the glucose molecule into ATP, the energy battery of the cell. On average, 36 ATP molecules are produced per glucose molecule that entered the cell. In the process of producing ATP, oxygen is brought in from the bloodstream to be the final electron acceptor in the electron transport chain, and the carbon dioxide that is produced as a byproduct is released. The goal of cellular respiration is to transfer the energy from the food that we eat daily into ATP that our bodies can use. This process starts with the eating of a snack or meal and ends with capturing the energy from the complete breakdown of the nutrients into energy and carbon dioxide.